I'm interested in what happens in a cell. So how can a cell decide or know that she's a muscle cell? How can a cell know that it needs to divide now to create a root precursor and a shoot precursor? So that's the kind of questions that interest me.
The first cell of a new organism is built by the fusion of the female egg cell and the male sperm. Fertilization brings together two sets of genes, one maternal set and one paternal set. Genes are written in the DNA and control all processes in a cell, such as division and differentiation. After fusion of the maternal and paternal sets of genes, this cell is the building block for a new organism, such as a human being or a plant.
During my PhD thesis, I was focusing on a phenomenon that is important for reproduction, both in flowering plants, where I worked on, as in mammals, namely genomic imprinting.
All genes are present in two copies, one copy inherited from the mother and one from the father. Usually both gene copies share the same fate, either both are active or both are silent. Having two copies of each gene serves as a backup. In the case where one gene copy is broken, the other copy is still functional and can substitute for the broken one. Whereas in most of the cases the two gene copies are equivalent, there are rare exceptions, the imprinted genes. For genes that are imprinted, only one copy is active. The other copy, while still functional, has been silenced. Imprinted genes are important for successful reproduction and are mostly active during embryo development. In mammals, imprinted genes are active in both the placenta and the embryo. In plants, however, imprinted genes were thought to be present only in the seed's placenta, the endosperm. They had not been discovered in the plant embryo until now.
So I discovered during my PhD thesis that genomic imprinting, the phenomenon I investigated, not only exists in the endosperm, which is the placenta of a plant seed, but also the embryo, which is kind of a novel finding that A, goes against the dogma and might find its way into textbooks. And second, it opens a lot of research questions about, how will this be translated into the next generation, what happens to those imprinted genes in the seedling, and so on. So the crucial thing to study early embryos and the phenomenon of genomic imprinting was to develop a method to isolate very early embryos.
Previously, researchers focused on embryos from 6 days after pollination. Michael, however, focused on embryos only 2 to 4 days after pollination. At this stage of development, the embryos are extremely small and difficult to handle. Michael and his colleagues developed an automated method to isolate the very early stages. This kind of fundamental research is only possible at an independent institution like the University of Zurich.
My biggest fear for the future is that science will not be free anymore. I consider science as a form of rational art, which is other than producing paintings, reproduce knowledge. And to produce knowledge in any kind of field, we need to be free. We need to be able to follow our curiosity and our fascination to try to answer the phenomena of nature. And to do this, we rely on public money, because corporate money is always aimed at applications, is always aimed at business value, whereas public money is aimed at creating knowledge, which should be the goal of fundamental research.